The 21st century has witnessed a revolution in biology with the advent of CRISPR-Cas9, a gene editing tool with unprecedented power and precision. CRISPR, short for clustered regularly interspaced short palindromic repeats, has the potential to transform medicine, agriculture, and our understanding of life itself. Unlike previous gene editing methods, which were cumbersome and expensive, CRISPR offers a relatively straightforward and affordable way to make precise changes to DNA. This breakthrough has democratized gene editing, making it accessible to researchers worldwide and accelerating the pace of discovery. The implications of CRISPR are far-reaching, touching upon fundamental questions about human health, food security, and the ethics of manipulating life's blueprint. As with any powerful technology, CRISPR comes with its own set of ethical considerations and potential risks. The CRISPR revolution is still in its early stages and its full impact is yet to be realized. CRISPR-Cas9 is a revolutionary gene editing tool that harnesses a natural defense mechanism found in bacteria. These microorganisms use CRISPR to protect themselves from invading viruses by targeting and destroying their DNA. Scientists have adapted this bacterial system to create a programmable tool that can target and edit specific genes in any organism, including humans. The CRISPR system consists of two main components: the Cas9 enzyme, which acts as a pair of molecular scissors, and a guide RNA molecule. The guide RNA acts as a homing device, guiding the Cas9 enzyme to the desired location on the DNA. Once the Cas9 enzyme reaches its target, it makes a precise cut in the DNA strand. Scientists can exploit these repair mechanisms to introduce specific changes to the DNA sequence. The potential of CRISPR in medicine is immense, holding the promise of curing inherited diseases, treating cancer, and even preventing viral infections. Scientists are exploring a wide range of applications, from correcting genetic defects in human embryos to developing new cancer therapies. One of the most promising areas of CRISPR research is in the field of gene therapy. Traditional gene therapy methods often involve replacing a faulty gene with a healthy copy, which can be challenging and inefficient. CRISPR, on the other hand, offers a more precise and efficient way to correct genetic defects by directly editing the mutated gene. For instance, CRISPR is being investigated as a potential treatment for cystic fibrosis, a genetic disease caused by mutations in the CFTR gene. Scientists are developing CRISPR-based therapies that can correct these mutations in the lungs, potentially curing or significantly alleviating the symptoms of this debilitating disease. Beyond its potential in medicine, CRISPR is also poised to revolutionize agriculture, offering solutions to some of the world's most pressing food security challenges. Scientists are exploring ways to use CRISPR to enhance crop yields, improve nutritional content, and make crops more resistant to pests, diseases, and climate change. One of the key advantages of CRISPR over traditional breeding methods is its precision. CRISPR allows scientists to introduce specific genetic changes into crops without altering other desirable traits. This precision breeding can significantly accelerate the development of new crop varieties with improved characteristics. For example, researchers have used CRISPR to develop tomatoes with increased shelf life, rice with higher yields, and wheat that is resistant to powdery mildew, a fungal disease that can devastate crops. These advances have the potential to increase food production, reduce reliance on pesticides, and improve the livelihoods of farmers worldwide. The unprecedented power of CRISPR to alter the genetic makeup of organisms has sparked intense ethical debates worldwide. One of the most contentious issues is the potential use of CRISPR to make heritable changes to the human germline, the DNA that is passed down from one generation to the next. While germline editing holds the promise of preventing inherited diseases, it also raises concerns about unintended consequences for future generations and the possibility of designer babies. Another ethical concern is the potential for CRISPR to exacerbate existing social inequalities. For instance, if CRISPR-based therapies are initially expensive, they may only be accessible to the wealthy, further widening the gap between the rich and the poor. Addressing these ethical challenges requires careful consideration, open dialogue, and the establishment of robust regulatory frameworks. The discovery of CRISPR has ushered in a new era of gene editing, one with profound implications for the future of medicine, agriculture, and our understanding of life itself. In medicine, CRISPR promises to revolutionize the treatment of genetic diseases, offering hope for millions of people living with debilitating and often fatal conditions. Beyond treating inherited diseases, CRISPR has the potential to transform cancer treatment, develop new antivirals, and even reverse the aging process. In agriculture, CRISPR offers a powerful tool to address food security challenges, enhance crop yields, and reduce reliance on pesticides. CRISPR-edited crops have the potential to withstand harsh environmental conditions, resist pests and diseases, and provide greater nutritional value, ensuring a more sustainable and equitable food supply for a growing global population. Scientists are exploring its use in a wide range of fields, from biomanufacturing to environmental remediation. The CRISPR revolution is still in its early stages, and its full impact is yet to be realized. The transformative potential of CRISPR is no longer a distant dream. It is already being realized in laboratories and clinical trials around the world. One remarkable example is the case of Victoria Gray, the first person in the United States to be treated with CRISPR for sickle cell disease. In 2019, Gray underwent a groundbreaking treatment where doctors collected her blood stem cells and used CRISPR to modify a gene that boosts fetal hemoglobin production. Gray's body began producing healthy red blood cells, effectively curing her of the debilitating symptoms of sickle cell disease. In another groundbreaking application, researchers in the United Kingdom used CRISPR to treat a young girl with a rare and aggressive form of leukemia. After traditional treatments failed, doctors turned to an experimental CRISPR-based therapy that involved engineering immune cells to target and destroy the cancerous cells. The treatment was a success, and the young girl's cancer went into remission. The CRISPR revolution is still in its early stages, and the technology continues to evolve at a rapid pace. Scientists are constantly developing new and improved CRISPR-based tools, pushing the boundaries of gene editing and expanding its potential applications. One area of active research is the development of more precise and efficient CRISPR systems. While CRISPR-Cas9 is the most widely used system, scientists are exploring other CRISPR enzymes with different properties, such as Cas12a and Cas13, which offer advantages for specific applications. Another focus of research is improving the delivery of CRISPR components into cells. Despite the rapid advancements in CRISPR technology, challenges remain, such as the potential for off-target effects, where CRISPR edits unintended parts of the genome. Scientists are developing new strategies to minimize off-target editing and improve the safety of CRISPR-based therapies. The advent of CRISPR has sparked not only a scientific revolution but also a profound societal shift, forcing us to confront fundamental questions about the nature of life, the ethics of manipulating our genes, and the future of our species. One significant societal impact is the potential for CRISPR to reshape healthcare. As CRISPR-based therapies become more widely available, they could revolutionize the treatment of genetic diseases, potentially eradicating some conditions entirely. This raises questions about access to these potentially life-saving treatments and the potential for exacerbating existing health disparities. CRISPR's ability to edit the human germline also raises profound ethical dilemmas. While germline editing could prevent inherited diseases from being passed down to future generations, it also raises concerns about unintended consequences and the possibility of designer babies. The societal impact of CRISPR is complex and multifaceted. The advent of CRISPR has ignited a profound debate about the future of human evolution. With the ability to edit our own genes, we are no longer passive observers of evolution but active participants in shaping our own destiny. This newfound power raises both exciting possibilities and daunting ethical challenges. On the one hand, CRISPR offers the potential to eliminate inherited diseases, enhance desirable traits, and even extend human lifespan. Imagine a future where genetic diseases like cystic fibrosis, Huntington's disease, and sickle cell anemia are eradicated, where humans are immune to viruses like HIV and influenza, and where our lifespans are extended significantly. On the other hand, the ability to alter the human germline raises concerns about unintended consequences for future generations, the potential for exacerbating social inequalities, and the very definition of what it means to be human. If we can choose the traits of our children, will we create a society where genetic advantages are concentrated among the wealthy, while others are left behind? The discovery of CRISPR has ushered in a new era of gene editing, one with profound implications for the future of medicine, agriculture, and our understanding of life itself. This revolutionary technology has given us the power to rewrite the code of life, to correct genetic defects, to enhance desirable traits, and to potentially cure diseases that have plagued humanity for centuries. CRISPR's legacy will be defined not only by its scientific breakthroughs, but also by how we choose to use this powerful tool. As we continue to explore its vast potential, we must proceed with caution, guided by ethical principles and a deep respect for the sanctity of life. The decisions we make today about the use of CRISPR will have far-reaching consequences for future generations. By embracing open dialogue, fostering collaboration, and proceeding with caution, we can ensure that CRISPR's legacy is one of hope, healing, and progress for generations to come.